Okay, so we're looking at antibiotics today, and antibiotics are one of the greatest inventions of mankind, but it's being abused and we have some problems here. Antibiotics are essentially a form of chemotherapy, so it's a chemical used as a therapy, and in this case, an antibiotic kills living things. In particular, antibiotics are only effective against bacteria. So, if your parents are telling you to take antibiotics because you've got the flu or you've got a cold, stop it. It will do nothing. Stop going to your dodgy doctors getting prescriptions for antibiotics when you don't need them. Now, antibiotics can be naturally derived compounds that we kind of see. Plants might produce antibiotics, or we might have them from molds and fungi, but some antibiotics are man-made as well, once we know a little bit about the pathogen. Because they only target bacteria, they're not going to really harm our cells most of the time. Some antibiotics have a pretty wide range of action, and in the process of killing bacteria, they might harm our cells as well, but these are like really, really strong ones, and they're not often used. With an antibiotic, we have lots of different modes of action. So as long as we know what bacteria we're dealing with, the antibiotic is going to stop that bacteria from growing and replicating. So it could either stop it by basically ripping apart its cell wall, it could stop it by stopping the bacteria from being able to replicate or double its DNA, or it could stop particular machinery inside of the bacteria that it needs to survive as well. Antibiotics were discovered by this guy called Alexander Fleming. Now, he gets a lot of the credit for this. Should he? I'm not so sure. There are other people who did a better job with what he discovered. Alexander Fleming was a bacteriologist working in London in 1928. He was known for not being the neatest guy, pretty slobby. So what he would do would be I'm doing an experiment, I'm gonna leave these agar plates full of this pathogenic bacteria open in the air, and I'm just gonna go on holiday or go for the weekend. Okay, bye. He did this a lot. He had a pretty messy lab, lots of things just growing anywhere. Kind of amazing that he fluked and found this new medication, but yeah, we'll get into that. One day, Alexander Fleming leaves these plates open, goes on holiday, comes back, and he realizes, oh, there's mold growing on this plate with bacteria. And he looks closer and he realized around the mold, you actually have a clear ring where the bacteria is not growing. This clear ring is actually known as something called a zone of inhibition. So it means that you've got something diffusing out into the agar that's killing bacteria and they're not able to survive. So Fleming was like, oh, this mold is producing something that's killing this bacteria. I wonder if I can research this further. Now, the mold itself was known as Penicillin notatum, and he grew this mold, had little bits of bacteria on there. Sort of what happened, notice it was always producing something that was killing certain types of bacteria. He kind of did some research on it, didn't really get that far, so he kind of gave up. So, resilience, not great. 10 years later, we have an Australian scientist named Howard Florey, and he was working with this German chemist named Ernst Chain. He looked at Fleming's work and went, there's something in here, we should definitely pursue this. So what they did, they extracted the chemical being produced by the penicillin mold, and they worked out that we could use this as a dose against bacteria. So what they did was they got 8 mice, and half of them were given this penicillin extract and half were not given this, but all were infected with a bacterial strain, Streptococcus. Now what happened was that the ones that were given the penicillin mold extract survived. Hypothesis supported. It wasn't all a success. When they tested this on humans, they gave a dosage to a man. They didn't get the dose right. He did die, so they worked out, we need to give him a bit more. But after that, we had a working antibiotic, the first in human history. Now this was a game changer. So what happened next? World War II. to. People dying left and right, disgusting, unsanitary conditions. But now we have this antibiotic that is killing a lot of bacteria. So what we had in the First World War is a lot of people died of blood infections, septicemia. In the Second World War, the numbers of this dramatically decreased. I mean, people died of other things in the Second World War, but like not bacterial infections. So thanks to all of their amazing work in 1945, Halbert Fleury, Ernst Chain, and Alexander Fleming won a Nobel Prize. Okay, so how does penicillin actually work? So when we look at pathogens, we had a little bit of a talk about bacteria and how there's a distinction between bacterial species, which is in their cell wall. Now, if you look at a bacterial species, they could either be gram-positive, which means that they have a thick layer of this protein called peptidoglycan, or they can be gram-negative, where they have a really thin layer of peptidoglycan. Those that are positive for peptidoglycan rely on this for stability and structure. Penicillin disrupts that. It basically stops the bacteria from being able to form this thick layer of peptidoglycan around their cells, which means the cell is really susceptible to lysis or breaking. So that means that this antibiotic is effective against any gram-positive bacteria. It's what we know as a broad-spectrum antibiotic. What's broad spectrum? A broad spectrum antibiotic is something that has a wide range of action against a lot of different species. So it's not super specific that it will only attack one strain of bacteria. So if you ever have an infection, it's really severe, but we don't have enough time for testing, a doctor might prescribe you with a broad spectrum antibiotic. Generally though, we like to prescribe narrow spectrum antibiotics, which are really specific in their action. So that means that they're only affecting certain strains. So why do we want to actually do that? A narrow spectrum antibiotic, it's less likely to kill the good bacteria that you have inside of your body. Remember, we need that to stay alive. If you have a broad spectrum antibiotic for a prolonged period of time, you're more likely to wipe out that good bacteria, and then you're going to need our old friend, the fecal transplant. Broad spectrum antibiotics as well have an increased risk of antibiotic resistance, and this is a massive problem that we're getting right now in the world. Now you've got to remember, bacteria replicate super quickly. They replicate exponentially through a process called binary fission. And every single time we're replicating, we have potential for mutations. So what we essentially are doing, if we prescribe antibiotics for the wrong thing, we're taking antibiotics when we don't need to, or if you're not finishing a course of antibiotics when you are prescribed them, is that you're giving bacteria a little taste of things out there that might be a challenge for them. Those that have a mutation to survive will keep surviving and keep replicating. So that means that the next time you take this antibiotic, you've got this resistant strain that's not going to die when it should. It's really, really strong, and it's going to keep going and cause a lot of problems. We're seeing really bad antibiotic resistant strains out there, and they tend to kind of hang out in hospitals because hospitals are like a disease factory. There's lots going on there that's really bad. A lot of immunocompromised people, so these pathogens are able to spread really, really quickly. The strains that we're seeing in hospitals is this thing called MRSA, which is known as methicillin-resistant Staphylococcus aureus. Long word. But it's essentially a strain of golden staph that's really resistant to this antibiotic called methicillin, which is quite widely prescribed. What this means is that people are getting staph infections, and it's really gross. Don't look up a MRSA infection because I will do that for you. Let's have a look. So, in this picture here, we can actually see this weird gold yellowish looking pus. Yeah, that's the golden staph growing in there. It's disgusting, and in this case what we can do is we can get rid of this tissue, basically strip away any of the dead tissue, a little bit of the live tissue as well, until we really make sure that infection is gone, and we're going to have to take an even stronger antibiotic. So we're fighting antibiotic resistance with stronger antibiotics, which means, who knows, they might mutate even more. So we've talked about antibiotics only being effective against pathogens such as bacteria. But that being said, if you've got a viral infection, it's not the end of the world. Not all is lost. We still have options to treat viruses. Now, what we might use is an antiviral. Now, we know a lot about how viruses work. We know how they get into a cell. We know what they do inside of the cell. So what we might do is we might block any mechanism of action of the virus to stop it from being able to replicate and burst out of your cells and start that infection cycle again and again. So the most common example that we look at is Relenza, also known as Tamiflu. Now, Relenza works by blocking this particular protein that we have on the surface of the influenza virus. So the influenza virus needs a couple of things. It needs to get into the cell using this protein called hemagglutinin, and then it's going to replicate, do its thing, but then in order to actually release itself from a cell, it needs this enzyme called neuraminidase to basically cut itself free from the host cell so that it's free to go and get into different cells. So what Relenza does is it blocks the active site, which is the cutting region of neuraminidase. And that means that the flu is stuck on the surface of the cell that's infected and it can't burst out and get into other cells. This is actually an example of this concept called rational drug design, which is when we know a little bit about our target. We know about the shape of certain proteins, or the shape of certain enzymes, and we're going to create something that's complementary to that shape to block it, which means it can no longer work. Rational drug design is a process that's created a lot of different drugs out there, and it's creating lots of different new drugs as well. So whilst we're looking at antiviral treatments, antibiotics, we are looking at treatments for diseases. These are not cures. They're things that basically slow down the progression of a disease so that your immune system has the time to sort of come in there and clean up everything and get rid of the pathogen properly. There's no such thing really as one cure. What might work in one person might not work in another person. So all that stuff that you see in the media of like, oh, there's a new cure for the disease, we're going to win against this new infection and nothing's going to hurt us. That stuff's false. So please don't believe the media, or in this case, don't believe the US president who talked about a cure for COVID-19 that was completely unfounded. So all this stuff that you see in the media, like, oh, scientists discovered a new cure for this disease. No, we've discovered a treatment. We've discovered something that might help to treat, but it's not a cure. Don't believe the media when you see that stuff. Don't believe American presidents that talk about a cure for COVID-19. Be smart. So thanks to antibiotics and antivirals, we have viable treatments to help support your immune system to take out pathogens before they can spread.